This lab covers the digestive system, which does contain a lot of histology, so make sure you take a look at the microscopes in the lab before the lab exam so you are comfortable with the way the organ looks as well as the specific structures within. Lab Objective 1 discusses the differences between mechanical digestion and chemical digestion. So recall that mechanical digestion has to do with the physical breakdown of food. So things like churning of the stomach, mixing in the stomach, chewing food. So taking larger pieces and breaking them into smaller ones. Whereas chemical digestion is breaking the bonds in the food. And so recall that these are organic molecules, so we're talking about primarily covalent bonds here. And then in terms of the end products of breaking down these different types of substances, what are the end products of carbohydrates? Those are monosaccharides, primarily glucose. With proteins, remember those are amino acids, and then with lipids, the most abundant form is going to be the triglyceride, which breaks down into glycerol and 3 fatty acids. So these are the components that will actually get absorbed into our bloodstream. So this is why mechanical and chemical digestion both have to occur during digestion. Before we take a look at the individual organs' histology, we can review the alimentary canal. Remember, this is a tube that is open on both ends, going from mouth to anus. So from the mouth into the pharynx, into the esophagus, into the stomach, small intestine, the duodenum, the ileum, and the duodenum, the jejunum, and the ileum, and then into the large intestine, ascending, transverse, and descending colon into the sigmoid colon and rectum and anus. Anywhere along that alimentary canal, we're going to see a very similar look of the histology. And additionally, there are accessory organs, the salivary glands, the liver, the gallbladder, and the pancreas, that are not part of the alimentary canal, but rather they produce secretions to facilitate digestion. So within the alimentary canal, there's the lumen on the inside, and that's the inside of the organ. The mucosa is the layer that lines the lumen, typically going to be epithelial cells. Beyond that is the submucosa, a lot of glands here, blood vessels, there's a nervous system network. And then the muscularis layer may contain multiple layers of itself. So for example, the stomach has 3 layers of muscle because of all the mixing and churning it does. And then lastly, the outermost row of cells has a couple different names. It's called the adventitia, it's called the serosa, and it's also called the visceral peritoneum. If you remember from general A&P, we talked about serous membranes and how there's always a visceral layer and a parietal layer. And the visceral layer lines the organ, and the parietal layer lines the cavity wall. So in this case, the outermost region of the organ is the same thing as the surface of the organ, which is the visceral peritoneum in this case. Lab Objective 2 starts our sort of look at the microscope slides and looking at the different types of organs and specific cells. So in all of these, you'll be looking at the mucosa, so the different types of epithelium, the muscle in the organs, and then also some unique structures therein. So if we look first at the esophagus is unique because it contains stratified squamous epithelial cells. Why would the esophagus want to have stratified squamous epithelial cells? Well, think about what the esophagus does, right? It's just a tube that transports food from the pharynx into the stomach. So there's a lot of friction that happens there. And so it's necessary to have multiple rows of cells. Remember that stratified refers to a multiple layers, and squamous is squished. And so that is going to have the best structure equals function relationship for the esophagus. You are going to want to know what the organs look like. So for example, the stratified squamous epithelial cells should be a trigger for you that is the esophagus. This is the only organ that we're looking at that will have that. Next, in the stomach, here's the lumen, and then the mucosa is sort of arranged in these almost wavy patterns. And what we're looking at are these dips called gastric pits, and the gastric pits will lead into gastric glands. And the rest of the mucosa is coated with columnar epithelial cells. Remember that columnar epithelial cells are long column-shaped structures. And a couple things about those cells within there. The different cells that we see in the gastric glands, which are going to be at the end of the pits, we're going to have chief cells, which secrete pepsinogen, fix that in a minute, and parietal cells, which are going to secrete hydrochloric acid. And what these are going to do is help to break down proteins. So proteins are digested by pepsin, but pepsin is the activated form of pepsinogen, and hydrochloric acid is necessary to activate pepsinogen. So the stomach, we're looking for columnar epithelial cells arranged in kind of these pits, and then these kind of puffy looking cells at the end of the pits represent parietal cells in the gastric gland. So the gastric gland actually contains the cells that make the gastric juice. Here's another picture of the stomach. Again, we see these little pits every so often in the mucosa, and that's going to lead into the gastric gland. So there's lots and lots of gastric glands here that we can see in the stomach. Again, because that primary role of digesting proteins, and that will help perform that. In the small intestines, there are a number of structures that we can look at, and one of them, if we look at the way the mucosa is arranged, we're going to see what are called villi. And villi, so villi is plural, and then villus is singular, so one villus, more than one villi. And these are finger-like projections of the mucosa. And one of the things about the small intestine is that it's so long, but each one of the cells and each one of the portions of the small intestine has structural modifications to make even more room to absorb nutrients. So the primary role of the small intestine is to absorb nutrients, So the villi, the intestinal villi, make up the mucosa, and then if we look at each villus a little bit more closely, they're also made of simple columnar epithelial cells. And when we're looking at these columnar epithelial cells, we're also going to see these little kind of footprint-looking cells, almost kind of look like bubbles or footprints, and these are called goblet cells. And goblet cells are cells that produce mucus. So when you look at most of these types of tissue here, the goblet cells with the mucus, that's going to help kind of protect the organs. If we go back to the stomach, we're also going to see goblet cells we're going to see not necessarily goblet cells, but cells that will make mucus as well, because the stomach acid will eat the stomach if it is not protected by mucus. We can also see the muscularis layer in the small intestine that's going to help to propel food through it. And again, simple columnar epithelium in this villus. What I want to do, I'm just going to fast forward several slides to slide number 14. This is going to show an illustration of a villus, or some intestinal villi. And what we want to see, so lab objective 3, again on page 45 in your lab manual, we're looking at the structures of an intestinal villus. So here's one villus, one finger-like projection. It is bordered with simple columnar epithelial cells and goblet cells. And if we look more closely at each one of these cells, it's coated with microvilli. And remember that microvilli are extensions of the cell membrane, and together they produce almost what looks like a hairbrush, so we refer to it as the brush border, and there's going to be a lot of enzymes in here for digestion. In addition to the columnar epithelial cells on the inside of the villus, we're going to see blood capillaries, again, for that absorption, and also the lacteal. And the lacteal is a lymphatic vessel. This is also going to help with the absorption of fats. And the lacteal, being a lymphatic structure, will help to clean the blood, so to speak. Most of the structural modifications in the small intestine have to do with absorption. So again, more surface area for absorption. And then if we go back up to where we were before with our small intestine, I'm going to skip large intestine for a minute and go back to it. One of the other modifications is called the Peyer's patches. And Peyer's patches are important because when food gets toward the end of the small intestine, it's pretty much feces. And so we want to ensure that the bacteria in the feces is not being absorbed into the bloodstream. So Peyer's patches we refer to as MALT tissue, which basically stands for mucosa-associated lymphatic tissue. And there are other areas of the body like the tonsils and the nasal cavity and oral cavity that also act as MALT tissue. And again, this is going to help to sort of clean up and make sure that bacteria is not entering the bloodstream. So if we go back to the slide we were on before, again, small intestines, look for goblet cells, look for the villus, look for simple columnar epithelium. With the large intestines, or the colon, this structure, by the time food gets here, again, it's mostly feces, and so most of the absorption that's going to happen is going to be absorption of water. So we are going to see simple columnar epithelium again, but rather than having all these villi, we will see a more sort of uniform structure, and that's going to give rise to the functions of the large intestine. So essentially, there's absorption of water here. There's also a lot of goblet cells, so kind of interspersed here, that will produce mucus. And that's going to help to lubricate and ease the passage of feces through the large intestines. Looking at the large intestines, let's look at this slide again. In the small intestine, again, these Peyer's patches are going to look like sort of these large circular structures in the mucosa. We looked at the villus, and so the next objective, objective 4, on page 45 of your lab manual, we are looking at the accessory organs, okay? So we're going to look at salivary glands, the liver, and the pancreas. And so the salivary glands produce saliva. That's their one job. And saliva will get produced in the salivary gland called the alveolar gland. See if I can move this down a little bit. And then get dumped into a duct. And then the duct will spit out, pun intended, that saliva into the mouth, into the oral cavity. So if you recall, this large salivary gland in front of the ear is the parotid salivary gland. Then there's one under the mandible, the submandibular, and then the sublingual salivary gland. And so each of those are going to have groups of cells within the alveolar gland, and the cells of the gland actually make the saliva. And then it's going to dump that saliva into the salivary duct, and that salivary duct will duct into the mouth. And so when you feel saliva producing, you are thinking about food, you're smelling food, you're seeing food, and basically that's prepping your body for digestion. Remember that the saliva produces salivary amylase, which is going to break down starches. And so we can see that chemical digestion will occur first thing in the mouth. Next, we have the liver, and the liver is a very important accessory organ. So it has a lot of functions, but in terms of the digestive system, it produces bile. And the liver itself, in terms of the organization, is made up of repeating subunits called lobules. And so this is what to look for when you're trying to identify the liver under the microscope. Look for these lobules, okay? It kind of looks like floor tile, a little bit with holes in it. And each one of the lobules is a hexagon shape and contains a hole in the middle for the central vein. If we look more closely at the picture itself of each lobule, we can see that there's little blocks of cells, and the cells are called hepatocytes. And then there are canals of blood vessels that link the blood vessels and link the cells, and those are called sinusoids. So we're going to see that blood coming into the liver is going to come through what's called the hepatic portal system, or hepatic portal circulation, and that's basically a kind of a bypass circulation of the main circulation, and it's so that the liver can do all of its nutrient interconversion and processing of bile and fats and all types of nutrients. And so we'll take a look at the microscopic structures, and then a little bit later we'll go back in and fill in these different blood vessels. But when we look at one lobule, again, we're going to see, if we look microscopically, here's that central vein, and then all the cells here are hepatocytes. And between the hepatocytes, there's little blood sinusoids. And if we go back to our picture, so all of these little sinusoids, which are here, are going to take that processed blood into the central vein, and then the central vein will ultimately empty back into the hepatic vein, which will empty back into the inferior vena cava. So again, what to look for are these hexagon shapes, which are the lobules. If we look at the pancreas, so the pancreas has several functions. It's an important organ, and it has an exocrine function, which will produce digestive enzymes, and it has an endocrine function, which will produce hormones. And there are two different types of cells that do this, and the exocrine cells are called pancreatic acini or acinar cells. One of those is called an acinus, so lots of terms there. And what we're going to see is that they both kind of look like different clusters of cells. And so looking first at the acini, these are kind of these circular shaped glands, and they are going to produce the digestive enzymes that we'll talk about a little bit later, and then dump them into the pancreatic duct. And then the pancreatic duct will dump into the duodenum. Remember that since the pancreas is an accessory organ, it's not technically part of the alimentary canal, but rather it facilitates digestion by sort of feeding into it. And so the acini look like circles, okay? They kind of look like these little donuts. Then you might see clusters of cells, and these are called islets of Langerhans, and these have the endocrine function. Within the islets of Langerhans, we have alpha cells, and those produce glucagon, and then we have beta cells, and those produce insulin. And glucagon responds to low blood sugar, so it's going to raise blood glucose. And insulin responds to high blood sugar, and therefore it's going to lower blood glucose. And so we're looking for these sort of islands or islets of cells. So microscopically, we will see those acini are going to be almost kind of these darker donut-shaped structures, whereas the islets of Langerhans are going to be these clusters of cells together. Now we can't tell which are alpha and beta cells on the microscope, but you should know that the whole thing is the islet of Langerhans, and it's made up of alpha and beta cells. A couple other pictures of the pancreas. Here's one kind of close up, where again we have these circular kind of donut cells. That's part of the pancreatic duct here and the acini, whereas these clusters of cells together make up the islet of Langerhans. Another picture again, the acini. And this picture almost looked like little, almost like cauliflower maybe, kind of lumpy. And then the islet cells again are going to be all clustered together. So remember that the acini have the exocrine function and the islet cells, alpha, beta, have the endocrine function. If we switch gears again, lab objective 5 is looking back at the liver. And so this is kind of showing part of the hepatic portal circulation. And so I'm going to kind of volley these different things together. But a closer up picture of one of those lobules of the liver. So each one of these cells is a hepatocyte. And there is a central canal or central vein and has sinusoids that flow into it. Coming into the liver, however, is the hepatic branches of the hepatic artery. Now this is important because this is going to bring oxygen-rich blood to the liver so that the liver actually has nutrients and energy for itself to use. And then the hepatic portal vein has brought all of the drained blood of the stomach, spleen, and intestines to get processed here. So the blood flow will ultimately dump into the central canal, and then all the bile formed by the hepatocytes will empty into a bile duct that will ultimately get back to the main part of the liver and then get stored in the gallbladder. What we can also see here is that the bile, I'm going to go back to that first slide that we looked at on slide 16. So we have those same structures here. Here are the sinusoids. Here's branches of the hepatic artery and the hepatic portal vein. And then these little green things over here are called bile canaliculi. So bile canaliculi are basically canals that connect cells and connect the bile ducts to basically collect all the bile that's being produced. If we go back to the other picture, one of the other types of cells we can see in the sinusoid is called a Kupffer cell, which is also known as a hepatic macrophage. And what this means is a macrophage is a big phagocyte, and phagocytes are basically scavengers looking for harmful materials. Lab objective 6, describing the function. So again, the salivary alveolar gland, that's going to produce saliva, and therefore salivary amylase. Pancreatic acinar glands, or cells, remember, is the exocrine function, so making digestive enzymes. What do the alpha cells do in the islet? Those produce glucagon, and the beta cells produce insulin. Hepatocytes are the liver cells. They're going to do the nutrient interconversion as well as produce bile. And then finally, the hepatic macrophages, the Kupffer cells, act as phagocytes in the sinusoids. Lastly, if we look at the actual hepatic portal circulation, so again, a portal is a separate blood circulation that's bypassing the normal and main circulatory system. So there's a lot of vessels listed here, but the ones I have in red indicate the ones on your sheet. So the blood that is draining from the spleen in the splenic vein, the gastric veins drain the stomach, and then the inferior and superior mesenteric veins drain the intestines. And all of those are going to drain onto this big vein here called the hepatic portal vein. And the hepatic portal vein goes into the liver. And then again, if we go back up 2 slides, here's a branch of the hepatic portal vein, and that's going to flow into the liver, into the lobules. And the liver will do its job of nutrient storage, breakdown, buildup, interconversion, produce bile, and then that blood will go back to the central canal and flow out of the liver, go back here, through the hepatic veins, and the hepatic vein goes back into the vena cava, inferior vena cava. So again, it's a way to bypass the main system. Lastly, for the enzymes, if you look at the table On page 8, or excuse me, on page 46, objective 8, it kind of breaks it down for you into enzymes for carbohydrate digestion. Those are going to be amylase and disaccharidases. Enzymes for fat digestion, that's a lipase, and then the rest are for protein digestion. And they're either going to come from the stomach, the small intestine, or the pancreas. And then one of them in the oral cavity in the saliva. So if you look at table 1 in your lab packet, there's a nice list there. And basically it shows you the enzyme, where it's produced, what it breaks down, that's the substrate, and then the site where it breaks it down. So for example, if you look at the first one, salivary amylase, that's produced in the salivary alveolar glands, and the substrate is carbohydrate, so it breaks down starches in the mouth. There are a number of cells in the small intestines, again, that contain microvilli, all of them do, but the brush border of microvilli produces peptidases and disaccharidases, and those, again, are going to break down sugars. So it'll be a good idea to, again, look through your packet. There's some really good information in terms of transportation and absorption. And there's also a nice flow chart on page 51 in your lab manual of those enzymes. So take a look at that as you are studying for this test.